Hi, I'm Abby. This summer I've worked in the Restrepo lab, and today I'm going to be talking about the good, the bad, and the ugly of the tomato pathobiome. Okay, so the story of my project begins actually before I even came to BTI in the previous summer. So during the summer of 2024, researchers from both the Restrepo lab and Zymtronics, which is a partner company, were in the field doing a trial and observed diseased tomatoes and diseased pigweed, which was nearby to the tomatoes, during a field trial. And so in order to figure out what was actually making the tomatoes and the nearby pigweed sick, the lab isolated bacteria and fungi from them. So Oscar obviously talked a little bit about the importance of the tomato, but it's an essential part of produce markets and processed foods. It makes up 1/5 of all vegetable consumption. And a prior study did show that in a plot with tomato and pigweed, when pigweed was allowed to grow for the entire season, there was a 68% reduction in tomato yield compared to the tomato plots without any weed at all, and an average decrease in weight per tomato by 25.9 grams. And so the story of the pigweed is very, very highly relevant to the story of the tomato. Okay, so a relevant question that comes up is, how did the tomato and the pigweed even get sick in the first place? So traditional plant pathology considers the disease triangle. And so the disease triangle said, okay, wait. Okay, I'm just gonna go back. Okay, so the disease triangle says that when you have the overlap of a susceptible host, a virulent pathogen, and a favorable environment, a plant is able to get sick. But what makes the Restrepo lab so unique is that it's interested in the pathobiome, which is the diseased microbiome of a plant, not just the microorganisms that are necessarily causing disease. And currently, the lab is working on describing the pathobiome of the pigweed. And so my project aims to complement the description of the pigweed pathobiome by describing the tomato pathobiome. That looks like first fully identifying both the bacterial and the fungal isolates that were collected, and then evaluating their pathogenicity. So to start off, I have 2 questions. The first question is, which fungi and bacteria are actually making up the tomato pathobiome? Second question, are all of the isolates pathogenic? Some of them, not any of them? And so to start off, I had to reactivate and actually grow up the fungal and bacterial isolates that were preserved from the previous summer. So this is what my fungi looked like and my bacteria. After that, I characterized those isolates by their morphology. And so just looking at slides of some of my fungal isolates, you can see that for 1.1a, there are conidia, and conidia are especially important for fungi because they're useful not only for asexual reproduction, but also for their pathogenicity. And then here, you can see a mycelium. And mycelia are more important just in the context of vegetative growth, occupying space on a plate. Once I looked at the morphology of the isolates, I moved on to describing them on the molecular level. And so first, I started with a genus-level description. So in order to start my genus-level description, I had to extract DNA from both the fungi and bacteria. For fungi, I PCR amplified and Sanger sequenced the ITS gene. For bacteria, I did the same thing with the 16S gene. The ITS gene and 16S gene are highly, highly conserved, which means that I can use one set of primers, universal primers, to amplify them. Super, super convenient. And then I'm actually able to make the genus-level differentiations. And so that looked like getting my forward read and reverse read. So forward primer, reverse primer. Obtaining a consensus sequence, and then actually taking that consensus sequence and comparing it to a database of strains of all different genera, and then evaluating the top hits with like the highest amount of query cover. And so as an example, you can see that for strain 7, for my 16S read, all of my top hits were the Pseudomonas genus. And so comparing my genus results to the pigweed genus results, you can see that there's some overlap. So both pigweed and tomato have overlap in the Pseudomonas genus, as well as Alternaria. But then they also had some genera that were distinct to each plant. But you can also see that this is a fair amount of isolates, especially if I'm zeroing in on a species level classification. And so I had to pick and choose which isolates I actually wanted to continue with. And so I chose Fusarium. Oh, okay. I chose Fusarium, Alternaria, and Pseudomonas, all of which have been known to cause symptoms of disease in the tomato, and also all of which the lab had previously been working on. And so then I was able to move on to both the species level description of the isolates and evaluating their pathogenicity via detached leaf inoculations. So species level identification entailed a second round of PCR and Sanger sequencing, except this time I was working with a set of housekeeping genes, so just pretty essential, pretty highly conserved genes for both bacteria and fungi respectively. And then it's also important to note that my primers were species specific rather than universal, which allowed me to make those species level distinctions rather than genus level distinctions. And then Sanger sequencing. But because I was working with multiple genes for both bacteria and fungi, instead of simply taking each gene and putting it into like an NCBI blast search, constructing phylogenetic trees allowed me to compile all the information for each of those genes in order to get higher resolution species level identification. So that looked like creating and curating a database of a set of strains for each of the genera that I had chosen, as well as an outgroup. So as an example, so for bacteria, GAP-A was one of the genes that I sequenced. So I'd locate the gene sequence for each of my strains in my database on NCBI, actually align the sequences, and then most importantly, I'd put the sequences for each gene together and then run a tree based on that concatenation. And so this is a complete view of the Fusarium phylogenetic tree. Obviously, it's very zoomed out. I don't expect you to be able to read it, but what I do want to point out is that way up here and way down here, I have two outgroups which do not have the genus Fusarium. They diverge the earliest on, which is validating the pipeline that I used to create the tree. And so zooming in a little bit, you can see my two Fusarium tomato isolates, 1.5b and 1.3. They're closest on the tree to Fusarium scurpii. It's also important to note that each of these branches have very, very high support. So Fusarium scurpii has been reported to cause symptoms of disease in tomato. And in line with that, with my detached leaf inoculations for both strains, I was able to see both sign of the disease and symptoms of the disease, so like necrosis of tissue for both of them. And so for next steps, I'd want to actually confirm that both my 1.3 and 1.5b isolates actually share the pathogenetic species identity with Fusarium scurpii. So that would look like re-isolating the fungi from the detached leaves, another round of PCR amplification and Sanger sequencing, and then adding them to the Fusarium phylogeny that I already created. And then interestingly, Fusarium scurpii has been reported as a soil fungus. But the bacterial and fungal isolates that I worked with from the summer of 2024 were collected from leaves. And so it also implies that there's some mobility to this fungus. So it's like able to travel up and down the hosts. Okay, and then this is a complete view of the Alternaria phylogenetic tree, also very zoomed out. Zooming in a little bit, so you can see 4.3, which is my first tomato isolate. Closest to Alternaria solani, which you might remember from one of my previous slides, has been shown to cause symptoms of disease in the tomato. And then interestingly, in this clade here, two of the pigweed strains are clustering together, which suggests that there is a strain of Alternaria that is perhaps unique to the pigweed that was nearby to the tomato. And down here, my other tomato Alternaria strain, 1.1a, is clustering with two of the pigweed strains. Interestingly, in my detached leaf inoculations, you could see symptoms of disease for each strain. And so for next steps, I would want to verify that that strain that was all by itself at the top of the tree, 4.3, actually shares a species identity with Alternaria solani. And then I would want to look a little bit more into those two pigweed strains that were in their own clade, because they serve as a potential avenue for weed control. And so in order to do that, I would grow the database that I used to create the tree by adding in more strains and the specific clade that those two pigweed strains were located in, rerun the tree to obtain higher resolution in order to draw like a more specific conclusion about the species identity of the pigweed strains. And then I'd also want to take those strains and inoculate them in both pigweed and tomato to see whether the Alternaria is actually able to survive on the tomato. And if not, then it could potentially be used to target the pigweed. Finally, given the large number of strains that were used to construct the Alternaria phylogeny, it is possible that the two pigweed strains that clustered with 1.1a, one of the tomato strains, represent a novel species. But in order to look more into that, I would want to re-isolate the strains and then add them to an Alternaria phylogeny with more strains in that specific clade. And then finally, if tomato and pigweed are sharing an Alternaria species, then it's possible that because they were close to each other in the field, the species is able to start in one and then move to the other. But again, I would want to perform cross inoculations to assess like potential host preference. Okay, and then this is my Pseudomonas phylogeny. So you can see that my 3 Pseudomonas strains are clustering with the pigweed strain, and that they're closest to Pseudomonas fluorescens, which interestingly is the opposite of pathogenic. It's actually been shown to promote both growth and dry weight of tomatoes, as well as to enhance their protection against pathogens. And then in my detached leaf inoculations, you can't see any symptoms of disease in any of the leaves. And so, noting that there's an Alternaria pathogen that could potentially travel between tomato and pigweed, it's possible that this specific strain of Pseudomonas could actually accompany that Alternaria, even though it's not pathogenic. And so in the future, I'd want to run antagonism assays between my Pseudomonas strains and Alternaria strains to test whether they're compatible. And kind of going back to this model of the disease triangle from the beginning, I learned from my project that it's not only important to consider the pathobiome of a susceptible host, but also the pathobiome of plants in their nearby environment, because like the reality is you might just be studying one plant, but that plant is never in isolation. There are weeds, there are other plants nearby. And so I think that considering the broader environment does kind of paint a more high resolution picture of a plant when it's sick. So thank you to everyone in the Restrepo lab. Thank you to Sylvia, my mentor Juliana, Yvonne for all of his help constructing the phylogenies. Thank you also to George, Delaney, and Megan for making the program possible. And does anyone have any questions?
So I know you did a lot of your work this summer. Was this for this project or for another project?
So it actually wasn't for this project. So I have a little bonus slide. So I actually was also involved with 2 other projects this summer. So the fieldwork was for a project called Leaves Are a Battlefield, which aims to perform both metagenomics and metabarcoding on the leaves of tomato, potatoes, and golden berries to describe the diversity of their microbiomes. But yeah, for that specifically, I was involved in weeding and also collecting some of the leaf samples. Yeah.
Since you used detached leaf pathogenesis like tests, do you think there's any difference between like if a bacteria is pathogenic on a detached leaf versus a leaf like or like have you tested if there's any differences between the levels of pathogens?
Okay. So I guess for people on Zoom, the question was whether there's a difference in pathogenicity of bacterial and fungal isolates on the field versus on detached leaves. And I would say, I mean, considering the fact that even with the tomato and pigweed, one of my conclusions was that isolates are potentially able to move from one to the other. I do think that it's important to take into account that like the microbiome of a leaf could shift based on its location. However, like the pathogenicity itself, I would expect, I personally would expect it to be most likely you'd get I would expect you to get the same result because it's the same leaf that you're working with. But I guess it is important to consider the fact that other microorganisms could be present on that leaf.
Do you think looking at the pathobiome of healthy leaves and kind of comparing against those that were diseased, could that be useful at all? I guess at identifying specifically those pathogenic disease-causing strains.
Yeah, so the question was, do you think looking at the pathobiome of healthy leaves could be useful at all in identifying the pathogenicity of specific strains? I think, yeah, I mean, I think that, well, first of all, I think just kind of coming back to the definition of pathobiome, at least the way the lab considers it is it's the microbiome of a leaf that is inherently diseased. But looking at the SONUS slide for leaves are a battlefield, one of the aims of this project is to describe diversity of microbiomes, not only with healthy leaves, but also with leaves if they potentially become diseased by a different pathogen, Phytophthora infestans. But I do think it kind of brings up the question of like potential shifts in like composition of microbiomes, like when a pathogen kind of enters the picture, and like in the context of that picture, I do think it's important to consider like the before, the during, and the after.
I've forgotten what the organism is, but I think it was in the first phylogeny you showed. There's one of the branches is like super long. I was wondering if you have an explanation for that. Is just like differences in sequencing quality or something like that?
Yeah. So the question was here. Let me try to go back. Okay. Maybe it was. Yeah, it was this one.
Yeah.
Okay. So the question was about the Fusarium phylogeny specifically, about potential reasons for the difference in length of the branches between strain 1.5b and strain 1.3. So I know I mentioned earlier that like for the bacteria and the fungi respectively, I sequenced like a set of genes. And it was very much the case that when I was amplifying and then sequencing, some of the genes were a lot easier to obtain like high quality bands for my PCR, high quality sequencing results than others. And so, yeah, I mean, oh, and that was another question that I was kind of considering when I was making the trees is like, it got to a point where my sequencing data was bad enough for some of the genes where like I was considering excluding them from the trees altogether because I didn't want them to actually influence the results. And in the case of Fusarium, I think I did include a little bit of sequencing data that was poor resolution, which could lead to the increased length.